So, hello, I'm Peter Buhl Jensen. I'm a CEO of Oncology Venture. I'm a medical oncologist and have worked for, you can say, translational medicine and drug development for many, many years in oncology. We have, I think, a new opportunity with technologies that allows us to read the genes and also how the cancers are operating. This gives us opportunity to, you could say, fingerprint the cancers to the drugs that are effective. And I was lucky to meet Professor Sten Knudsen in my previous company, TopoTarget. We had 2 products approved in TopoTarget. We met Professor Knudsen when we were developing product number 2, and we were struggling to identify, it worked, but we didn't really know where it would be best to go. And he said, why don't you go into peripheral T-cell lymphoma? And that was based on his big data analysis going all the way back to 2008. And he was right. This is where that product was approved eventually in TopoTarget. I started working with Sten Knudsen in 2011 and working on identifying, you can say, opportunities where our, you can say, the match that we work on could work. And we now have, I think, we actually sort of try now to use this word cure when we talk about this. This is about that giving the right products to the right patient at the right time gives us opportunities to cure patients. And out there are many cancer drugs, like belinostat we developed in TopoTarget. There are many cancer drugs that are developed, and only 5% of them actually enter the market because there's a lot of problems. I mean, a lot of them don't work. But even products that are very effective, they may be stopped because you don't know who should have this product. And this is where our technology is interesting. We can identify the patients that are likely to respond to a number of different products. And if we can get those up and we can actually have success in, you don't have to be able to see tumors shrink in all the patients. Many of those hard to treat cancers, products can be approved if they work in only 1/4 of the patients. And when those products are approved, it's obvious that there is also a high return for the investors on that. Those products usually cost more than $10,000 in the US and in Europe per month. And what we have is a system for, you can say, a companion diagnostic technology that allows us to look into patients' tumors to match with drug products. And then we buy in products where this system is able to provide, you can say, the solution to who should have the product. So it is really, if we could pick the right patients to our products, we would have a higher success rate in drug development and we'd be much more cost efficient. And this is, you can say, it's almost like pathway identification, matching the product with the patient's tumor. And then you can say there are, in essence, we now know that there's like sort of 6 hits. That's what happens whenever we get a cancer. It's like, you can say the accelerator is being stuck, speeder, and in the car, I don't know what you call it in Swedish, the accelerator, it can be stuck 100 different ways in cancer. And the brakes can break down, you can say, in 30 different ways. And the cells may not start to commit suicide as they should be doing. So it's like there are thousands of variations. And what we read is, how is this cancer operating and how is this matching with our products? And it is possible. We can actually measure in patients' biopsies. This is a patient biopsy, a breast cancer patient biopsy. It's put into paraffin so that the pathologist can cut it. And we can look today with modern technology. This is what everybody can do, but our match is to basically identify the algorithms, who should have which product. And this is then who should have the treatment that we are working on. So we have in-licensed now 6 products in our pipeline that all have demonstrated efficacy in patients, demonstrating that the tumors shrink, but the problem has always been who will benefit from those products? How can we identify who will benefit? And we have 2 products that are in liposomes, that is a new administration of old, very effective products, cisplatinum and doxorubicin, in new, you could say, clever liposomes, where we have proven in practice that it works, and we are developing those products. So we have several different products, all, you can say, united by being active, but also being united by we need to know who should have those products. It is very nice. In fact, it is rocket science. I think this is cutting edge science, and what we are doing is what sort of traditional, and this is what we start with always, is that we have a drug in a cancer cell. This is a square cancer cell here, and we are treating it with a drug, and the drug and the target is interaction, and something is happening. This is how we usually do our biomarkers. What Sten Knudsen would do, he's a systems biologist, he's a statistician. He looked at panels of cell lines, say what is actually the variation in sensitivity, because it may be that this works, but sometimes it doesn't even get in because the drug may be pumped out, or there may be some signals that is killing the whole thing that we hope for, so that the cells will not die even though there is a drug-target interaction. So he was able to come up with a much broader systems biology view, adding a lot of information into those algorithms. So we usually work with 200 or 400 different genes when we talk about which patient should have this product and which patient should not. So it's really like, you can say, the old pathway where we like to look at pathways in our school books, but in reality it's much more complex. We may have different 600 proteins that are involved in deciding whether a cancer cell is going to die from your product or not. So you can say the difference is that we now work with this big data-driven prediction, but also that Sten Knudsen saw that. He formed his first company in 2004 and built this system and has been, you can say, stubbornly working on that until now. And it's not now, before 2019, that we're actually doing prospective studies and show that he's right. So I'll give you an example. This is a liposomal formulation of cisplatinum. A smart liposomal, it actually is going into the tumors so that you are getting a release of the cancer drug to the tumors and not so much into the normal tissue. And then we are adding our response prediction to this product. So you all know about cisplatinum. This is one of the products that has cured sort of many patients. For instance, Lance Armstrong, he got testicular cancer in 1993 and was cured for his testicular cancer by cisplatinum. We also have an earlier example here in Denmark. This is Ole Ritter. It must be very dangerous driving those bikes. He also got testicular cancer and was cured by cisplatinum. So this product is one of the most effective products, but it's not curing breast cancer patients. It only works in 10% of breast cancer patients. It works in lung cancer, but it doesn't cure too many. So we have to say, how can we use this more elegantly? And it's a 50-year birthday now. This is a publication in Nature in 1969 on cisplatinum. So it was invented sort of 50 years ago. And you start with these terrible pictures. This is rats with a tumor, and you see the controls, they all died. But then you gave these rats down here, they got cisplatinum and they were cured. So this is how the products come into. We don't know who should have that product, but we certainly can see we can cure some rats with cancer. And then you put it in your first patient and you say, well, I don't know if it's going to work with you, but it worked in rats. Are you willing to test it? And Ole Ritter was one of the first actually, and he said yes, and he was cured. So he was a lucky guy there. So you can say the thing is that cisplatinum is so broad. So there's actually a homepage where they call it penicillinum cancer. It's of course a little too much, but in essence, Sten Knudsen, he made his company in 2004 basically trying to put much more information into the response prediction, and at the same time we have this. I mean, this product's been on there for years, and here we have Fojo and his colleagues working in 2005 saying, well, you know, platinum compounds, they have a lot of intracellular targets, and they have a lot of mechanism of resistance, so it's difficult to see who should have the product. And then we have Sten Knudsen's work, and basically this is a 2012 publication, Oncology, and again, that, there's so many different mechanisms of cisplatinum resistance, even though it works in testicular, ovarian cancer, head and neck cancer, colorectal, bladder, and lung cancers. It's an important product. We just don't know who should have it. A year ago, or in 2018, we published that we have now molecular prediction in cisplatinum in 2 different lung cancer populations here. We were able to demonstrate that. And basically, if you're not sensitive to platinum, your survival is very low. If you are highly sensitive, you get a very high likelihood that you'll survive your lung cancer chemotherapy that you give after surgery to those patients. So we say this is interesting. We now have a very strong platinum predictor. Let's get a product, a new formulation of platinum where we can use this. And this is the liposomal platinum that we have here. And we're using, you can say, the same data. We're using the same algorithms as we used in the lung cancer study with platinum, but now we have a much more smart formulation, gets into the tumors, and we want to treat breast cancer patients, but not all of them. It's all about selecting your patients. We treat 1/3 of the patients. Just for comparison, Herceptin, which is one of the most successful products out there ever, is being used in 25% of patients. They are the ones that are HER2 positive. It's 25%. So if we take top third, we might be also, it's okay with this. It's a very frequent disease. And what we've seen now is that we have 10 patients who in this top third, and we have 10 patients with this that are, when we look at the biopsies, they're very likely to respond. We don't want to treat those patients down here with low likelihood, but these ones. And when we have those, we're getting a very nice response rate. We've seen 5 out of 10 where the tumor has shrunk to more than half size. And that is basically heavily patients that have received 7 previous therapies. They've received a lot of other therapies. And this, in some of the cases, this is the first time where the tumor is actually shrinking. Not that the other products didn't work, they just kept the tumor, you can say, calm, but we're actually reducing it to half size. The last product that was approved for metastatic breast cancer in this setting was eribulin from Eisai. And when you look at how many patients did they see a similar shrinkage, it was in 12%. So if we are comparing now, we say, how many patients do we actually need to include in a study where we're comparing with eribulin? It's very limited. So now we're talking to the FDA saying, can we have an approval with, say, 100 patients? If we are able to keep a response rate of, say, even 30, 40, 50%, wouldn't it give us an opportunity to have this product approved? This is the discussion with the FDA. Here you see how it looks when we look at the middle 1/3 compared to the top 1/3. So we didn't include anyone here, but in the middle, it could also be that it worked. But you see those patients, this is weeks before the tumor comes back, starts growing, and we only have, you could say, 8 weeks before those patients. It's not good enough to those patients. But in the top third, it takes 25 weeks. These patients have received a lot of previous therapies. So 25 weeks in this setting is very important. It's half a year, and they're getting, you could say, an opportunity to live. We think they live. It gives us an opportunity to live longer. We can, in a way, we can compare because those patients received 7 previous therapies. So we can say we got 25 weeks with our product. And what did the patient receive the last time? The treatment that she just received before she received our treatment. And here we see a 14.5 weeks time to progression. So that means that there's a 10-week window. It's a new treatment option for those patients. They can, of course, get all these other products. But number 8 product may, at this late stage in their cancer, may give them a half year longer survival. We, of course, want to put this earlier so that patients would get it when they're not as sick, when they're not full of those tumor cells. But even at this time point, we can do something. So this is an example. This is another product that was approved with 117 patients just here in November. This is how we like to see the development, but also that because we want to do a single-arm study, we just want to show the response rate, we'll be able to tell the market how is this developing, and this is what we aim for. So you can say in 2021, we could have our product approved in the US first. And what is the value of that product? You can say for us, it's a $500 million. If we look at the risk-adjusted net present value, we're talking about that. And if you look at Herceptin, if this is, I don't think it's a Herceptin, but if we're getting there, Herceptin in 2007 sold $7.5 billion. This is a huge, huge market. If we can do, of course, if we can have breast cancer patients surviving longer, if we can get this product into lung cancer, head and neck cancer, or the other cancers that we're looking at. So this is what we aim for. This is, we looked in lung cancer with our products, cisplatin, but we've also a lot of other products where we have demonstrated in prospective and retrospective studies that it actually works. So this is why we're buying products into our pipeline, because we've shown repeatedly that we're able to predict with several products. So we have not only a Liplacis, some of those products down here, we don't want to do a 100-study trial. We want to take 10 patients in, 20 patients in, and demonstrate that we can double the response rate compared, or triple the response rate compared to what it was before. But that's the aim with those different products. So we are interested in, you can say, hamstering products in that where we can identify responders and do small trials. Liptosis, we can now see that we can have it approved with 100. I'm not saying that we'll do that ourselves. We're looking for partners. Of course, we're looking for partners also for liptosis. So we do a lot in breast cancer. That means that we can call these kind of umbrella studies. So we're doing the same screen. So we're looking with the same gene expression on these different products. And it gives us an opportunity to, you could say, look at the metastatic breast cancer. We have now screened 1,600 patients in Denmark with metastatic breast cancer. And when there is an opportunity for a product to be tested, the patient's going to be asked. Of course, we have asked all those patients if we could look into the biopsies, and they have approved it. I think there was one out there who said no. I mean, not everybody knows that the biopsy is actually at the pathology department. So it's also something that you don't need a new needle in the patient. It's not that you need a new biopsy. You can use the old biopsy. And this is how 800 patients, and we say thank you, 800 Danish breast cancer patients, how are they actually the different? Every vertical here is a patient. And here you can see how they differ in their likelihood to respond to our different products. And so blue is not very likely to respond, red is likely to respond, and then you say, okay, this patient could respond to our refolvin or could to our PARP inhibitor or whatever. There's always something in our pipeline that could be of interest to the patients. That's that.